Hello students. Today we will be studying about carbohydrates part 3. We are going to learn how to draw Fischer projection and Haworth projection of aldose. So this video is specially dedicated for aldose. Next video we'll try to learn how to draw the Fischer and Haworth projection for ketose. Okay. So today we are going to learn how to draw Fischer and Haworth projection of aldose. Now I guarantee you, when you see the complete video, you will be able to draw the Fischer and Haworth projection for the given carbohydrates. So let's begin. So first of all, let us understand what do you mean by Fischer projection. So before trying to understand or draw the Fischer projection, first of all you should know the basic definition of carbohydrates, which are already done in our part 1. Okay, so I'll just recall the definition of carbohydrates. Carbohydrates are nothing but the organic compound containing carbon, hydrogen and oxygen. So here, how you are going to represent the carbohydrates? As carbohydrates basically are the ring structures, but the same thing if you show it in the open format, okay, we can show the carbohydrates in open format using Fischer projection. Now what do you mean by Fischer projection? We can represent the molecule in two-dimensional structure. Okay, that is on paper. Actually all the organic molecules or all the molecules are actually having three-dimensional structure in space. That is spatial arrangement. But the same thing when we draw it on the paper, okay, and if you represent them using the lines, that is vertical and horizontal lines, then we call such projections or such structures as Fischer projection. So what are Fischer projection? Fischer projections are nothing but the molecules which are drawn in the two-dimensional way, that is this way. Okay, let us represent this structure where there are 4 atoms and there are 2 bonds. Okay, bond 1 and bond 2. One is vertical line or vertical bond. This is your vertical line, or we can call it as a bond. Okay, and this one is your horizontal line. So here what does Fischer projection indicates? Vertical line represents the bonds and the atoms in the plane, whereas horizontal line and the atoms represent out of the plane. Okay, out of the plane. So here out of the plane, or in the definition I have shown that it is in the page and out of the page. So horizontal line along with the atoms, that means this horizontal line and the atom, this one atom and this one atom, they will be out of the plane. One will be above the plane, one will be above the plane, and this will be below the plane. Okay, whereas these three, that is red ball, your bond and green ball, all will be in the plane, that is the plane of the paper. Okay, so this projection which we have drawn here is known as Fischer projection. What is this projection known as? This projection is known as Fischer projection, which is showing you two-dimensional structure. Now the same thing when actually in the space, how it will look? It will look this way. Okay, the same structure which we have shown with the balls and the lines, it will look like this. That is, let us label this structure as structure 1 and this as structure 2. So structure 1 is the actual structure which we will be seeing the space. In the space, suppose the carbon has 4 atoms, and out of those 4 atoms, one atom is below the plane, that is this blue one. It is because it is represented by dash and wedge. This is your wedge. Okay, wedge indicates above the plane. So here your ping ball and a rod or a bond will be coming in front of your eyes. That is, it will come out of the whatever screen we have. So it is going to be out of the plane, whereas blue ball and the dash is going to go behind the screen. Okay, so this atom is going to go behind the screen, behind the page or screen we can say, whereas red ball, green ball and the carbon atom, all these will be in the plane, okay. This way, this is our original molecular structure, and this is how second structure represents your Fischer projection. So in two-dimensional way we can represent this three-dimensional structure. Okay, so let us see the example. This is your simplest carbohydrate, which contains 1, 2 and 3. Okay, in MCQ also I have asked this question, that which is the simplest carbohydrate. So the simplest carbohydrate is of 3 carbon chain, which is called as glyceraldehyde. So this is a structure of glyceraldehyde, where if you consider this second carbon is a chiral carbon, as you can see all the groups attached to second carbon are different. Okay, they all are different. One is OH, one is CH2OH, one is hydrogen and other is CHO. So all the 4 groups are different. That's the reason why we will call such carbon as chiral carbon. Chiral carbon. So at chiral carbon you will find we can show the Fischer projection. Okay, we can show the Fischer projection where first carbon, second carbon and third carbon along with the groups. Okay, so this group, then third carbon chain group and the center one, all are in the plane, whereas your OH group and H along with the bonds will be out of the plane, okay. So this projection is your Fischer projection for the first carbohydrate. This is the simplest carbohydrate we have drawn and we have shown the Fischer projection. So I'll just remove whatever circles I have done. Now this is how the projection will look like. Okay, so this is known as Fischer projection. So let us see a few more examples how to draw the Fischer projection for the higher carbohydrates. Now before we go to the next part, let us understand the carbohydrates can be drawn as D and L configuration. Okay, the new concept which you are going to understand here for carbohydrates is Fischer projection only we will be drawing, but in D and L configuration. So what is D and L configuration? Let us understand that. Suppose here your pink ball indicates OH group. Okay, what I am saying, let us say that pink ball is indicating OH group. I'll write it here, OH. Okay, and this one is H, let us understand. Then this may be CHO group, and this green ball indicates CH2OH. So this is your glyceraldehyde structure. Okay, so this is structure 1 and this is structure 2. Now here when we are seeing a carbohydrate in a Fischer projection, both these diagrams, that is structure 1 and structure 2, we have drawn it in a Fischer projection. Correct? Because Fischer projection indicates what? A plus sign structure, vertical line and horizontal line. So this is a Fischer projection where OH, for considering D and L configuration, we have to concentrate on what? We have to concentrate on the second last carbon. Means we will be counting from the last carbon. Now here if the first carbon is aldehyde, second carbon is your middle one, third carbon is your CH2OH. Okay. Similarly here first carbon is your CHO, second carbon, and third carbon is CH2OH. So here which is the second last carbon? Second last carbon is your second carbon. Okay, because there are only 3 carbon. If there is a long chain, then second last carbon you have to consider, means from below the second last carbon. Now from below if you see the second last carbon, it is your second carbon. Now from on second last carbon you have to just find out where is OH placed, whether it is on the left hand side or whether it is on the right hand side. So here in the first structure OH is on the left hand side, means this indicates that first structure is your L configuration. L configuration of the glyceraldehyde. So this will be labeled as, the first structure will be labeled as what? First structure will be labeled as L glyceraldehyde. Okay. Similarly the next one, second structure, because OH is on the right hand side. See here OH is on the right hand side. So it will be labeled as D. Right hand side OH indicates D configuration. So D configuration will indicate it is a D-glyceraldehyde. Okay, and this way you can write for all the carbohydrates. This was the simplest carbohydrate we have considered in a Fischer projection, that is glyceraldehyde. So you have to see for D and L configuration which carbon, we have to see the second last carbon. Second last carbon, that also OH group, where the OH group is placed, whether it is on the right hand side or left hand side. So once again we'll recap. When the OH is on the right hand side, we call it as D configuration, and when OH is on the left hand side, we call it as L configuration. So these are the structures of your glyceraldehyde, D and L. Okay, so let me just erase this. Yeah, so here you can find D configuration I have shown on the screen. Similarly L configuration of glyceraldehyde. Okay, so you can count the number of carbons and then write down. This is first carbon aldehyde group, second carbon, third carbon, where on the second carbon OH is on the right hand side, so it will be labeled as D, and OH is on the left hand side, then we will label it as L. And these both are, L and D are both isomers of each other, or we also call it as enantiomers, because they are mirror images of each other but non-superimposable. They are mirror images of each other but non-superimposable. So they will be known as enantiomers. So let us see few more examples of D and L configuration of carbohydrate. Next is 4 carbon chain. If you see, next example which we are going to consider is 4 carbon chain. The previous example was glyceraldehyde was 3 carbon chain. Now if you see this example which is on the screen, you can see it is 1. We will always start labeling from aldehyde. So first carbon is aldehyde, second, third and fourth. The last one you will see mostly in carbohydrates, and that also aldose. Okay, because this video is dedicated to aldose, and you know what do you mean by aldose. Aldose means carbohydrate containing aldehyde group. Okay, those are known as aldose. So here first in aldose always the first carbon is aldehyde, and the last carbon chain in the chain, last group will be CH2OH. Okay, so student, you have to always remember this, that in carbohydrates, that also in aldose, we have already classified carbohydrate in our previous videos. Carbohydrates are classified into different saccharides, monosaccharide, disaccharides, polysaccharides. Okay, and they are further classified into aldose and ketose. Now these aldose, this is whatever example we are seeing today, those are all example of aldose. So let us see 4 carbon chain aldose. 4 carbon chain aldose is what? The name is called as erythrose. 4 carbon chain erythrose and D. So what does D indicates? Second last carbon. Which is the second last carbon? That is C3 carbon. C3 carbon, the OH is on, hydroxyl group is on right hand side. Right side. That's the reason it is called as D, and the name of 4 carbon chain aldose is known as erythrose. In erythrose you will always find both the OH groups are on the same side. Okay, both the OH groups are on the same side. Now if you see L erythrose, see here L erythrose is exactly the mirror image of D erythrose. Okay, L and D erythrose are mirror images of each other. But you don't have to consider the second carbon, okay, for labeling it as L or D configuration. We have to only see the second last carbon, that is this carbon. You have to check out where is the OH. Here in the second last carbon, OH was on the right hand side, whereas here OH is on the left hand side. That's the reason why this is called as L configuration. So in erythrose you have to remember that OH groups are on the same side of the bond, okay, whereas for 3 O's, if they are on the opposite side, the OH group. Now this is again the 4 carbon chain only. If you see 1, 2, 3 and 4 carbon chain of aldose, where OH groups are on the opposite side. Okay, the chiral carbon, these are your chiral carbons. These were your chiral carbons. Okay, on chiral carbon, if you see the second last carbon, where OH is placed on the right hand side. So this is the OH which we have to find out where it is placed. It is on the right hand side. That's the reason why we are writing it as D. D erythrose. And erythrose indicates that OH groups are on the opposite side. Okay. So see the L erythrose again. L erythrose, you will find the OH are on the opposite side. Erythrose means trans, yeah, anti OH. Okay. In my college days, I used to remember in this way: 3O cis and erythrotrans. T indicates trans. This is how I used to remember it. So you can also remember in the same way. Trans in 3O indicate trans, that is OH are on the opposite sides. Now L and D will represent whether the OH is on the left hand side or right hand side. If it is on the left hand side, then we will call it as L erythrose. If it is OH is on the right hand side, we will call it as D erythrose. I think this is clear to everyone, student. So you have to, I'll repeat once again, D and L configuration can be only considered considering the second last carbon OH group, where it is placed, whether it is on the right hand side or whether it is on the left hand side. Okay, so we have seen the Fischer projection. Now these all whatever projections we are seeing here of carbohydrates or aldose, they all are Fischer projection. Why Fischer projection? Because the whole structure we are showing it on only vertical and horizontal lines. Okay, here a vertical line represent what? In the plane. Horizontal line indicates out of the plane. So let us go to the next one. So in this we will be seeing about aldopentose, where aldopentose indicates what? Aldo is aldehyde and pentose is 5 carbon, C5, okay, of carbohydrate. So let us see the Fischer projection of aldopentose, and in this screen we will be, or in this slide we will be seeing only D aldose, okay, that is D configuration. Now I have shown some 3 structures here, but how to draw the aldopentose, let us first understand them. Before understanding the how many structures we can draw of aldopentose, let us understand the formula. The aldopentose is what? A carbohydrate having 5 carbon chain. In that first carbon as we have shown here, will be aldehyde group. First carbon will be aldehyde, then second carbon, third, fourth, and last carbon will be CH2OH. Okay, and the remaining here they may be OH, 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 H, H, and H. So this is the structure of aldopentose containing 5 carbon chain. 1, 2, 3, 4, and 5. Okay. So this structure which we have drawn here can have various isomers. It can have different isomers. What do you mean by isomers? Isomers having same molecular formula but structure is different. So how will you guess that aldopentose has how many isomers? So for that you have to know the formula. What is the formula for isomers? Knowing the isomers of carbohydrates, it is 2 raised to n. Okay, the formula is 2 raised to n. And what does n indicates? N indicates the chiral center. Chiral center. Now in this structure, okay, let me draw this again here. The first structure, first one. This is your OH, 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 H, H, and H. And the first structure name is D-ribose. What is the name of the first structure? D-ribose, where all the OH are on the same side. Now if you see the number of carbons, 1, 2, 3, 4, and 5. Okay. Now how many chiral centers are there? N indicates what? Chiral center. So let us see the first one. Is it chiral? No. Second one? Yes. Why? Because all the bonds which are there for this carbon, you will find they are different atoms or different group of atoms attached to this. So the second carbon is chiral, third carbon is chiral, and fourth carbon is chiral. Fifth is not a chiral. So what is chiral center here? N is equal to 3. Now if n is equal to 3, the formula becomes 2 raised to 3. So if it is 2 raised to 3, it is 2 into 2 into 2. That means 2 to the 4, 4 to the 8. 8 isomers we will be getting for the molecular formula this. That is a carbohydrate containing 5 carbon chain. Carbohydrates containing 5 carbon chain. So here how many isomers we should get for aldopentose? We should have 8 isomers. Out of these 8 isomers, we will have 4 enantiomers. Okay, 4 enantiomers we will be having. Means 4 pair of, we will have 4 pair of enantiomers. What do you mean by pair? Pair means if one is D, the other one will be, the other enantiomer will be L. Okay, 4 enantiomer indicates what? D and L enantiomers. So that's the reason why we are going to show not all 8 structures, but we will be showing only D configuration of aldopentose. Because D configuration means how many isomers we have to show? I have to show 4. First structure we have already drawn. Then we will be drawing second structure. Okay. Then third structure and fourth structure. So this way we will be drawing 4 structures or 4 isomers, enantiomers, that also of D-aldopentose. Only D configuration we will be drawing. And what did I tell in the previous slide? D indicates that OH is on this, OH is on the right hand side. So second last OH, let me draw every structure OH on the right hand side. So if OH is on the right side, hydrogen should be on the left hand side. So now once I have drawn OH on the right hand side, means all these structures, structure 1, 2, 3 and 4, all are what? Having D configuration. We are going to draw all D configuration. Okay, D configuration. So this way we will be showing the aldopentose. Now let us draw the OH groups. Now if OH group of second carbon is on the left hand side, whereas the remaining are on the same like राइबोस के जैसे ही थर्ड और फोर्थ कार्बन का OH राइट हैंड साइड पे है, सिर्फ सेकेंड कार्बन का OH अगर लेफ्ट हैंड साइड पे है, then the name of this compound becomes what? It is arbinose. It is called as D-arbinose. So I will write it here only, arbinose. Okay. Then third structure we are going to show in this way, that OH groups are on. Now third carbon OH we will be shifting and remaining we will keep as it is. So what we have done, we have only changed the third carbon OH group from the first structure, that is from ribose. All OH were on the right hand side. Now what we have done, only third carbon OH we have shifted to the left hand side. So it has become isomer because space arrangement will be different, and it is an enantiomer of the first ribose. So name of this compound is D-xylose. Okay. Similarly, fourth, one more enantiomer we can show here of our aldopentose, that is aldehyde group CH2OH. And last one, what we have to do is we will be showing for second and third carbon OH on the same side, whereas fourth carbon OH is on the right hand side. Now this is our fourth enantiomer. If you see all the structures, they are neither mirror images nor superimposable. Okay, they are neither mirror images, so they can be even called as diastereomers. Okay, because these are all means first structure, second structure, third structure and fourth structure, all are there diastereomers. Now why we are written here enantiomer? Because its L configuration will be mirror image of this L configuration. Whatever we have drawn are D configuration. If I draw L configuration, D and L will be mirror image of each other, but they are not superimposable. That's the reason why we have written enantiomers. So how many enantiomers we will get? Total we will get 4 pair of enantiomers. So I have drawn first part of the enantiomer. Second part will be your L enantiomers. Okay. So let me just remove this. You have to remember this formula, students, for finding out how many isomers are there. So isomers we can calculate using 2 raised to n. N indicates the chiral center. Okay. Depending on how many chiral centers are there, you can get those many pair of enantiomers. Okay. So if there are 8 isomers, that means 4 enantiomers we will be getting. 4 pair of enantiomers we can draw. So let us see now this I have shown here L. In L what we can do? You can just change the positions. Exactly the mirror image of ribose. So mirror image of ribose if I draw, it will be L ribose. Similarly, mirror image of second structure if I draw, it will be L arbinose. Okay, and so on. Now what will be the mirror image? If I draw the mirror image, my OH will be on this side. Similarly, OH will be here. Then H, OH, H and H. So second structure and this structure will be mirror image. Similarly, first structure OH is on the right hand side, OH we have to draw it on the left hand side. And here OH, OH, H and H. So this structure will indicate what? This structure will indicate L ribose. And so on, you can go on drawing them. Okay, all 8 structures you can draw. 4 for D and 4 for L configuration. So these are the Fischer projections for aldopentose. Similarly, let us see for aldohexose. Now you are clear what are hexose? Hexose means having 6 carbon chain. Carbohydrate containing aldehyde with 6 carbon chain. Okay, those are called as aldohexose. Now student, before we draw with the isomers, tell me how many isomers it will be having. 6 carbon chain will have how many isomers? Any guesses? Yes, what is the formula? The formula is 2 raised to n. And in this 2 raised to n, let me show here before 1, 2, 3, 4, 5 and last sixth one is CH2OH. So if you try to find out the chiral centers will be only the middle one, leaving your first and last carbon. So how many carbons are there in between? There are 4 carbon. All will be chiral. Okay, all will be chiral carbon. So what if you substitute this, means total in hexose, how many chiral carbons are there? 4 chiral carbons are there. 4 chiral carbons are present on hexose. Okay, that means n is equal to 4. So if n is equal to 4, isomers will be 2 raised to 4. Okay, isomers will be 2 raised to 4. That is 2 into 2 into 2 into 2. So if you multiply them, you will be getting 16 isomers. Total how many? For this structure we can draw 16 isomers. Okay, again isomers having same molecular formula, structure will be different in the space. So 16 structures we can draw for aldohexose, out of which if you talk in terms of enantiomers, how many we can have? How many pairs of enantiomers? Obviously the half, that is 8 enantiomers we will be having. 8 pair of enantiomers we will be having. That means 8D configuration and 8L configuration. Okay, I think now this is clear to everyone how to find out the enantiomers. Now we will just go and draw the structures. We'll try to draw the structure very fast now because I have clear your concepts of how to draw the Fischer projections for 5 carbon chain. Now we are going to see for 6 carbon chain. Okay, that is aldohexose. Let us be little fast, otherwise my video will be very long. So let us see. Okay, these are the 8 which you can just write down the H and OH groups and you can get the structures. Now let me do it in the fast way, where OH, okay, OH, second carbon, third carbon, OH and OH. If all OH are on the one same side, okay, then it is called as Aldose. The same way, if I only change the second carbon OH group, whereas all other are on the same side, that is this, then it is called as D-ultrose. Now all these configurations are of D. Okay, I will not write it again and again because you can see second last carbon having OH on the right hand side. Okay, second last carbon OH is all on the right hand side. So all are D configuration. All 8 isomers which I have shown here are what? They are all D configuration. So I have written on all the 8 isomers OH on the right hand side. Now depending on other things, now the third structure we will draw of glucose. This is a glucose structure, very important. You have to remember the structure of D-glucose because it is, whenever the questions are asked, they are based on glucose or fructose. Okay, so in aldohexose they ask about D-glucose. So here let us understand how the OH groups are placed. OH, H, OH, H and OH, H. So here your third carbon position of OH and H has been changed. Here the second carbon OH was changed. Okay, so this is how the position of third carbon is changed. Now next one is mannose, D-mannose, where you will find that the position are changed for first and second carbon chain. Other two remains the same side. Okay, these have been changed. That is D-mannose. Then it is next is gulose, D-gulose, where you will find the OH of 1, 2, 3, 4th. Fourth carbon OH will be changed. The other will remain on the same side. Only fourth carbon OH has been changed. Okay. So this is which is called as D-gulose. Then idose. Here you will find the OH are alternate. So if second last carbon OH is on the right hand side, then here it will be left hand side, then again right hand side, and again left hand side, and this structure is called as idose. Okay. Then next is gala-lactose. Again D structure because OH of the second last carbon is on the right hand side. So gala-lactose, you will find that the OH of third and fourth carbon are on the same side, whereas second carbon it is this way. Okay. And the last structure, last isomer of hexose is your talose, where what is the change? Only your second last carbon OH is on the right hand side, all remain on the left hand side. Okay. So this way we have seen all the Fischer projections for D-aldohexose. Okay, D-aldohexose. So students, just take a screenshot and practice these structures. All the structures you just practice them on the paper, then only you can remember them. Okay. So let us go to the next part. I think you have taken the screenshot. Write it down on your paper and see the structures. Okay. Let us go to the next. So student, today we will be seeing in this part, we will be seeing Fischer projection to pyrenees. Now what we are going to see, up till now we have seen all Fischer projections this way. These were the Fischer projection with vertical and horizontal line. Now we have to convert them into Haworth projection. That is Haworth structure. Okay. So Fischer projection, we are now going to study, understand how to draw the Fischer projection into Haworth projection. Now there are two Haworth projection we can draw. One is called as pyrenees, another is known as furanees. Okay. Both are basically Haworth projection is nothing but ring structure. As we have seen, Fischer projection was open structure. Fischer projection is nothing but open structure, whereas Haworth projection is a ring structure. That means we will be getting a closed ring. Okay. Now these closed rings or Haworth projection, we can draw it in a two ways. That is pyrenees and furanees. Pyrenees indicates what? Six-membered ring with oxygen. So considering oxygen in the ring, there is six-membered, like this it is. Okay, the oxygen will be also present in this ring. So if you count 1, 2, 3, 4, 5 and 6. So such type of six-membered ring if you get along with oxygen, it is called as pyrenees. And if you get five-membered ring with oxygen, along with oxygen, then it will be called as furanose. Furanose indicates 5-membered oxygen ring. 5-membered oxygen ring. So this will be called as furanose, because you know that this is furane and this is pyrene. So with oxygen 5-membered ring, then it is called as furanose. Okay, furanose, and this is pyranose. So these projections are known as, when we draw such carbohydrates in this fashion, which will indicate 3-dimensional structure, it is called as Haworth projection. It will indicate Haworth projection. Okay, so let us begin to draw the Haworth projection. So basically you should understand how to first draw the pyranose and furanose. Then we can easily draw the Haworth projections. Okay, so let's begin with pyranose forms a form of D-glucose is 6-membered ring, oxide ring. The aldehyde group at first position whatever is the aldehyde group, and OH on the 5th carbon are going to be involved in this formation of pyranose. How it is? Let us see. So this is how it is going to form. The first structure which I have shown here is our D-glucose. You can count the number of carbon. First carbon aldehyde, last carbon CH2OH, and the middle carbon, that is second, third, fourth, fifth, are having OH groups. Only third carbon OH is on the left hand side, all other are on the right hand side. So it is a D-glucose. Okay, now when you are going to convert it into pyranose, see here what is happening. There is 5th carbon, what we have said, first carbon and OH of the 5th carbon are going to make a ring. Okay, are going to make a ring. How it is? This way. Oxygen has lone pair. The lone pair of 5th carbon OH group is going to attack on the aldehyde. Okay, it is going to attack on the aldehyde. How? This way, on the carbonyl of aldehyde, and your carbonyl group I bond of O, C double bond O, is going to take the proton. Okay, and it will form, it is going to take a proton and it will form OH, and this hydrogen will donate electrons to oxygen, because oxygen is going to form a bond, and from here this proton will be removed, which will be attached on the first carbon. Okay, OH. So it forms OH. Now you can see this structure which we have drawn. If you count the number of carbons and oxygen, they will be 6. So let us count from here. First carbon 1, 2, 3, 4, 5, and this is your 6th oxygen. Okay, 6th oxygen. Obviously for the straight chain, 6th carbon was the last one, but if you are going to make a ring like structure, the 6th is your oxygen. Okay, so here I have shown one star. What does that star indicate? That your the aldehyde carbon, that is first carbon, first it was not a chiral center, but once your oxygen electron pair had made a bond with first carbon, it has converted into a chiral carbon. Okay, chiral carbon. Such carbons are known as enantiomeric carbon. Enantiomeric. Okay, they are known as what? They are known as enantiomeric carbons. So enantiomeric carbon, both are enantiomeric carbon. Then you might see that in both the structure, both are 6 carbon chain. Okay, 1, 2, 3, 4, 5, and this is 6th one. Both are same, but one in first structure, that is your, let me say this is second structure and this is third structure. Now in the second structure you will find that the OH is on the right hand side I have shown, whereas in the third structure OH is on the left hand side. Now when this oxygen lone pair is going to attack, it may attack from this side or it may attack from this side also. So depending on from where it is going to attack, we are going to get two isomers. One is alpha and other is beta. Okay, it is alpha D glyco. Why glyco? Because the structure we have got it from glucose. So glycopyranose indicates 6-membered ring. So D-glucopyranose, but one is alpha and other is beta. So here alpha indicates that the OH group is on the what? Bottom side, and beta indicates OH is on the top side, okay, upper side. So if beta is there, beta indicates what? Upper side OH group, and alpha indicates below side or below the plane OH group. So alpha indicates below the plane OH group, which OH only anomeric carbon we are talking, okay. Alpha will tell you about the OH group of anomeric carbon, the carbon which are just converted into chiral center, okay. That carbon is called as anomeric carbon. So if OH is below the plane, it will be known as alpha. If OH is above the plane, then we will call such structure as beta, okay, such structure as beta. So this way you can draw the you can convert the Fischer projection into pyranose. Now next which we are going to see is about the furanose. In furanose, if you see, furanose are nothing but, I have already explained that 5-membered ring with oxygen. So for same D-glucose, if D-glucose we are going to convert it into 5-membered ring along with oxygen, that is oxide ring, then it will be called as furanose, okay. Furanose is simply 5-membered ring with oxygen. So of aldose. Now here let us see the D-glucose, and D-glucose can be converted into furanose. Like we want 5-membered ring along with oxygen, so obviously the 4th carbon oxygen should attack on the aldehyde group, okay. 4th carbon hydroxyl group should attack on the 1st carbon aldehyde group, that is this way, okay. So here when 4th carbon OH group attacks on the 1st carbon to make it as anomeric carbon, okay, it may attack from front side or back side, depending on that. You will be getting 2 isomers, so one is alpha and other is beta. Because we are getting 5-membered ring, 1 2 3 4, and this is 5th carbon, 1 2 3 4 5th carbon, sorry, not 5th carbon, 5th oxygen. So this ring shape is going to make your what? Beta D glucopyranose. Now let me show the actual Haworth projection of this, okay. Haworth projection of both the things, let us draw. One is about pyranose and one is furanose. Now in furanose you will find that the structure will be this way. We'll draw here only the Fischer projections, sorry, Haworth projection. Okay, those are ring shaped. So let me show one, that is beta. I'll show beta 1. So this is your 1 2 3 4 and 0, that is oxygen, 5th one. Now your first carbon, always whenever we are showing anomeric carbon in Haworth structure, it will be always on the right hand side. So we are always going to show the anomeric carbon on the right hand side, okay, of the chain. Now here in beta structure, what I told you, beta structure, the OH is always at the above the plane. So you have to show OH at the top, means above the plane, and H below, okay. So this will indicate what? Beta structure or beta isomer. Now let us see, if OH we have shown above the plane, then second carbon where it was from this beta structure, OH here it was on the left hand side, second carbon OH is on the right hand side. So exactly opposite we have to draw here, H OH. Then third carbon OH is where? It is on the left hand side. So again OH H. Then here this one we have already shown. Now this is your 4th carbon. 4th carbon, what are the things which are there? H is there and this whole group is there. Now what you have to do? You can show H here and the whole group. What is the group on 4th carbon? Here it is C, COH, H and CH2OH. Okay. So this way, I'll just remove this Haworth projection which I've written. We'll write it again. So this way we can draw the Haworth projection, that is Haworth projection or structure of the closed ring structure. Okay. So this is going to indicate your beta glycopyranose. It is beta glycopyranose. Same way, only if I change the position of this OH group. Okay, position of OH group. Now if I write the OH group down, then it will become alpha. So you have to just observe the carbon which is next to oxygen on the right hand side. That carbon will be your anomeric carbon. Anomeric carbon, the OH is on the bottom, then it is alpha. OH, if it is on the top, then it is beta. Okay. So I think this is clear to everyone. I'll just draw the beta only first, because if you're going to watch the video as I've shown with blue the arrow, so here it should be OH on the top, which indicates beta form. So I think this is clear to everyone. We have seen the furanose. Similarly, we can also draw pyranose also with 6-membered ring in the same fashion of Haworth structure. So this is what Haworth projection we are going to show for D-glucose. Okay. So here I have drawn one D-glucose. I have shown the pyranose as well as D-glucofuranose. So this is your 6-membered ring, and this is 5-membered ring. Okay. If you see here, pink ball indicates OH group, green ball indicates H. This is your oxygen. Yellow ball indicates carbon. So you can see here there are how many? 1, 2, 3, 4, 5 and 6. Okay. So all green ball indicates what? Indicate hydrogens. Yellow ball indicates carbon. Pink ball indicates OH and so on. So you can just write it down here: OH, H, OH, OH, H, H, OH and H. So this is your D-glucose. D-glucose we can convert it into pyranose. So which carbon OH is going to attack? Obviously 5th carbon, so that we will be getting 6-membered ring in this fashion. And if 4th carbon OH is going to attack on aldehyde, then we will be getting furanose. Okay. So these both are your D-glycopyranose and D-glucofuranose. Now tell me, which is it, a alpha or beta? Yes, can you guess? D-glucopyranose, is it alpha form or beta form? So if you pay attention, this is your oxygen and this carbon indicates what? Anomeric carbon. Anomeric carbon. So as I mentioned here, the pink ball indicates what? Pink ball indicates OH group. Okay, pink ball indicates OH group, means OH group here is at the bottom. So if it is at the bottom, OH is at the bottom of the ring, then it is alpha. Okay. Similarly, furanose also, here you can see from oxygen, this carbon is your anomeric carbon. The OH is again at bottom. OH is at bottom. So it is again alpha form. Alpha form. So both these structures are basically alpha and alpha D-glucopyranose, alpha D-glucofuranose. Okay. I think now this is clear with you. Now you, as I had promised, that when you start seeing the video, you will be able to write both Fischer as well as Haworth projection. And I guarantee you that I'm sure that you can now write both the structures, that is Haworth and our Fischer projection. Now this is, so here we are going to see the Haworth projection for D-ribo ribose, and that also furanose we are going to draw. So ribose are carbohydrate containing 5 carbon chain, 4 and 5. Again, green ball indicates hydrogen, pink ball indicates OH, yellow ball indicates carbon, and red ball indicates oxygen. Okay, this is how we are going to represent. So the same way we will draw it here: OH, 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 H, H and H. So here these are both equivalent, one in the atomic form and other, both are Fischer projection of D-ribose. Now D-ribose, if you want to convert, what we are saying, we are converting into Haworth projection of furanose. Furanose indicates what? 5-membered ring. What does furanose indicate? 5-membered oxide ring. Okay, so if I want 5-membered oxide ring, which OH is going to attack on aldehyde, so we have to count 1, 2, 3, 4, and obviously fifth should be oxygen. So fourth carbon hydroxyl group has to attack on the aldehyde to get a Haworth projection. Okay, so let us draw the Haworth projection for this. So here, if this is my fifth, that is oxygen is fifth, then right hand side, what I told you, it is always an anomeric carbon, that is this first carbon. So here OH, let me draw first the OH on the bottom. Okay, OH, if it is on OH, H means if when the oxygen is going to attack on the aldehyde, if whatever OH is going to form, if it goes below the plane, okay, if it is going to go below the plane, then let us see this, how we are going to show the remaining things. Okay, remaining, that is here you can show OH, H of second carbon, then again because all OH are on one side, so we can show all OH on the bottom side of the plane. And this one, let me draw it with the blue pen. 1, 2, 3, 4, and fifth will be here. Fifth carbon will be here. That is your this carbon. Okay, here we have to show the fifth carbon. Now what is that fifth carbon? It is CH2. So CH2OH. So this is your fifth carbon. I'll rewrite it again properly. So make a little longer. It is CH2OH. And this is your fifth carbon. Okay, fifth carbon. So here if you see the anomeric carbon, the OH is on the below the plane, that is bottom, below the plane. So my structure here, it is alpha form. What is my structure? It is alpha form. Similarly, I can draw beta form of D-ribofuranose. Simply what I have to do, I have to just change the position of OH, of anomeric carbon. If my anomeric carbon OH is at the top of the plane, above the plane, that means above the plane, then what you will be getting? You will be having a beta form of D-ribofuranose. So I guarantee you now that you can write both the projections, that is Fischer as well as Haworth projection, as well as I have made the concept clear of 5-membered ring as well as 6-membered ring, that is pyranose and furanose, and how to write the alpha and beta structure using Haworth projection. Okay, so students, I think you must have liked this, and now I guarantee that you can draw the structures of Fischer and Haworth projection for carbohydrates. Okay, now if you like the concepts, whatever I have taught you today in this video, please do like the channel, subscribe it, share it with your friends, and do write the comments that you like the videos, so that I will get the motivation to do such videos more for you and clear your concept. Thank you. Keep watching and Happy learning. Bye till then.